Scientists in the US say they've developed a new gene-swapping technique to prevent babies from inheriting diseases. So, is it a step towards healthier humans or designer babies? And should scientists be allowed to interfere with nature? This is Inside Story. Hello and welcome to the programme. I'm Sami Zaidan. Well, it's a procedure that could prevent children from inheriting diseases such as strokes, blindness and deafness, kidney failure, even heart disease. Healthy embryos are being made using 2 women and 1 man. Scientists are trying to eliminate defects that affect an estimated 1 in 4,000 children. But it's stirring that ethics debate. A lot of questions about the morality of having babies with 3 parents, the implications for the child's descendants, and the prospects of babies to order. Well, let's look at the procedure in more detail then. A woman's egg cell contains thousands of what are called mitochondria, each with its own DNA. And if they're defective, they can be passed on from mother to child. Well, the egg also has a nucleus. This holds chromosomes, and it's these that influence traits such as eye colour and height. Well, the new technique involves removing the nucleus from the mother's egg cell, which might have some defective mitochondria, and inserting it into a healthy donor egg cell. A sperm cell is then injected to fertilise the egg, which is re-implanted into the mother. So, is science creating so-called designer babes? Well, to help answer the question, we're joined by our guests in London, Josephine Quintavalle. She's Director of Core Ethics, a public interest group focusing on the ethics of human reproduction. And joining us via Skype from Chicago, Laurie Zoloth, Professor of Bioethics and Medical Humanity at the Feinberg School of Medicine at Northwestern University. And in Manchester, we've got Brian Bigger, Senior Research Fellow at the University of Manchester and a specialist on genetic therapy. Well, Brian's worked extensively on gene therapy and neurological disorders. Good to have you all with us. Brian, if I could start with you. First of all, help us to put this into perspective. Will this technique allow us to eliminate all hereditary diseases or just some?
Well, unfortunately, it's not going to allow us to eliminate all genetic diseases. However, there are a significant proportion of our genetic diseases
how significant the
from
proportion?
Defects in the mitochondrial.
How significant a proportion? What are you talking about? 50%? 90%?
It's probably about 1 in 100 of genetic diseases, single gene defects, arise from mitochondrial DNA mutations. And these are the first category of disorders that we would be able to treat.
All right, now let's perhaps move the discussion on to Laurie. When we talk about eliminating a significant % of hereditary diseases, are we talking about the most dangerous, the most important ones?
Mitochondrial diseases are important. They're only now beginning to be understood. That's not my concern when we think about this therapy. No one quarrels with the fact that it would be very good to get rid of mitochondrial diseases. Is this the best way to do that? What other effects does this cloning technique have? And this is the kind of technique that challenges 2 very serious ethical bright lines that we've erected to regulate this kind of technology.
All right, it sounds like you've got some doubts. What are they?
Well, one bright line we erected was when we thought about gene therapy, we discussed it in the following way. We said, you can alter the genes of an individual person, but we ought not alter the genes that are permanently passed down to the next generation and the next and the next, because we're so uncertain about the long-term effects of that kind of intervention. So it violates that bright line, because of course it's passed down to the next generation. The second bright line is we've said pretty much with every single discussion we've had about cloning technology that we can't use cloning to make human babies. It's just not safe enough. We don't know the long-term effects. And swapping out parts of the egg cell is a complicated systematic process, and the effects are unknown and may not show up for many generations.
All right, I want to bring Brian in. Hang on, let me bring Brian in. Do you agree with that? Are we passing on, are we making changes we don't really fully understand with this technique and passing them on for future generations?
I think we certainly have to move with caution at this stage. it's very true that we have the potential to pass on genetic changes to the children and the children's children beyond that. And so I think that this point is actually well made, that we need to be quite careful. I mean, another aspect of this was that Mitalopov and colleagues who developed this technique in Oregon actually found that although it worked very efficiently in monkey egg cells, that they had problems in about 50% of the human embryos or the human eggs that they were able to alter using this technique actually turned out to have genetic abnormalities. So I think we're still quite a long way from actual clinical application.
All right, but should the research be allowed to continue with caution, as you're saying?
Personally, I think it should, and part of the reason is because they're actually using an unfertilized egg at this point.
All right, hang on, Brian. I'll bring you back in. I want to go to Josephine here at this point. We've got a very cautious scientist on board who says, look, I've got some concerns too, but we need research to go on. Is he wrong?
I think he is wrong, and
why?
His last comment. He's wrong because he's not. We should be cautious, but we shouldn't go on. I mean, he's focusing on the egg, and I'd like to get back to that. Where do these eggs come from? I've looked at this research. There were 7 women involved, all healthy, and they produced 15 eggs per cycle for this research project. That in itself is abnormal. What we're doing here is doing a massive number of abnormal things to try and get rid of abnormality. Now, everybody would agree that we should try to find cures for disease. It's how we go about it. You don't get eggs easily. Women have to have their ovaries stimulated, and sometimes they suffer as a result of that. So I think that is a consideration right from the start. But if you look further into this research,
but if they're happy to do it, does that make it okay? If they're happy to do that, and is it a case where you can weigh up the pros and cons and say, you know, if this can overcome things like multiple sclerosis, let's have a shot at it?
Well, I don't think you can have a shot at everything without considering all the ethical implications. As I say, of course we've got to cure disease. The only way that you can find out whether these systems work is by further experimentation, implanting them in other women and seeing how the embryos develop. And I think that's something that even if a woman puts herself forward for it, we should consider that that's an absolute ethical no.
All right, I know that Laurie is really trying to get in here, but before we come to you, Laurie, I want to go back to Brian, because we heard from Josephine there that she said, you're wrong. You shouldn't be allowed to go on with your research because you're going to get into very muddy ethical waters whichever direction you go. And perhaps you shouldn't even have gone this far. Your response?
Well, this isn't research that we're personally involved in. We actually concentrate on gene therapy for children, in fact, and we're looking at other rare neurological diseases affecting the brain. Right.
However, perhaps I should clarify. I mean, you as a representative of the scientific community. Let me put the you in that context.
So I would come back to Josephine by saying that, you know, in the case of mitochondrial diseases, it's very clear that it's going to be a very long time before we can treat babies with mitochondrial diseases, adults with mitochondrial diseases, really to any level of efficacy, because gene therapy in adults is really pretty ineffectual, except in very rare cases. And so I think that where you have the opportunity to offer a mother an option not to pass on a mitochondrial disease to her offspring, then that should be offered as a possibility.
Could I say very simply, keep on with your research, and I hope you find cures for children with mitochondrial disease. But in terms of using other women's bodies, using their eggs for these purposes, one could say simply, why doesn't the woman then with the mitochondrial disease simply accept a donated egg from another person without trying to manipulate and create this absurd distortion of nature where you have you're going to create a child who has 2 maternal genetic parents and a paternal. I mean, there's another study going on in the United Kingdom where they're considering combining 2 embryos from 2 different sources, where you'd actually end up with a 4-parent child, 2 fathers, 2 mothers. I think cures have got to be acceptable.
All right. I want to let Laurie into the discussion.
Give me a
Laurie,
chance here.
please
so what
go
I
ahead.
think I'm agreeing with my colleagues from the UK. I think, and in fact, the UK has a model system for considering this kind of research and involving the public, and it's created a very robust debate. We have ways to do that in the United States, and they haven't been tried by these scientists. They simply publish the paper. Now, to do basic research on cells in a dish is one thing. To do research in primates, in chimpanzees, seems to me to be acceptable to work out this technique as a theory. But that's a long way from asking an individual woman to become pregnant with one of these very highly manipulated embryos. My colleague is correct that it needs the participation of the women to donate eggs, to get hyperstimulated through excessive hormonal stimulation, to create a great number of eggs, for a woman to become pregnant. And of course, there's the reality of a child that might be born out of this technique that might have far greater disease than the original mitochondrial disease. There is much more low-tech
but
ways.
hang on, Laurie. If researchers can find a way to reduce some of those risks, would you be on board?
I wonder about the use of technology that uses cloning technology. This is the thin edge of a very large wedge, and it's one that we've said we don't want to have happen. We don't want the kind of ethical fallout that cloning technology would create. We don't want.
So your objection is more ethical rather than scientific, or the risks that we might generate other problems, correct?
There's significant ethical concerns about the process involving the women, the other women, the use of eggs, the harvesting of eggs. There's significant safety issues that the UK has pointed us to, and everyone who's looked at this technology, every scientist has said this would be very difficult to make us stable, to make it stable. 50% of the eggs, as our colleague noted, were significantly mutated in ways that could be quite harmful or fatal. So the final considerations about what cloning would mean for us as a society, we really debated it for a long time, and we've decided to turn back from that technology.
All right, let me bring Brian back into the discussion. 2 points, Brian. First of all, about finding a way to eliminate all these potentially harmful side effects from this technique. Is there a way to do that?
I think that's going to be difficult. I mean, there are actually two techniques that were developed. One in the US, and one, as Josephine and a colleague in London pointed out, by Professor Doug Turnbull in Newcastle in the UK, which involves pronuclear injection. So that would be injection of genetic material into a fertilized egg in that particular case. In the UK, in fact, the Human Fertilisation and Embryology Authority has actually put this process out to consultation to the general public. And they're essentially letting the public make their mind up as to whether they feel that this is an acceptable technique to research further. And I think we are still a long way from making this a safe technique. You know, clearly, it's very clear from the research that's been done that monkey cells are not the same as human cells. That they were able to achieve a much higher level of efficacy in the monkey cells than they were in the human cells, and a much lower level of genetic abnormalities turning up in downstream.
Okay, so it's not really 100% safe yet. But the other point I want to put to you is about ethics. A lot of the objections are based on ethics, and we've heard things like, do we want a kid with two mothers and one father? And I'm sure you're a human being with ethics and morals as well, but you're pro at least carrying out some more research at this stage. So how do you answer that question when people say, we don't want people with 3 or 4 parents? It's not something we're comfortable with as human beings, is it?
So I would say to you, go and meet some of the parents of children with some of these rare diseases and talk to them about whether they want a treatment for their children. And they will tell you very strongly that they will do everything in their power to get a treatment for their kids. You know, in this case, I agree that there are certainly ethical concerns about how the eggs have been obtained for the study. But it's possible then in the future that those ethical concerns could be addressed. And I think, you know, a public consultation process is the way forward. It's very important that we have that debate.
All right, let's remind everyone a little bit about the study findings that have been attained so far. The research is being conducted at Oregon Health and Science University in the US, and the procedure still needs some federal approval to be tested in human beings, of course. But here's what the scientists have achieved so far. Well, in 2009, they created eggs with DNA transplants from Rhesus monkeys. 4 developed into healthy animals. Then the team has now used the same technique to create human embryos, with about half having abnormalities though. But they say some 20% of the eggs did produce embryos that would have been suitable for transfer back to the mother. All right, let's perhaps take things to Laurie. I mean, one of the researchers on this technique says you can expect the first healthy baby to be born using this technique in the next 3 years. Are we really that close, or is that a bit of propaganda?
I would not be happy if in 3 years this technique had been rushed through discussion. We need a much longer time to think about this. You know, it's gene therapy, and I'm on the Federal Recombinant DNA Advisory Committee that looks at gene therapy. We've been looking at techniques and ideas, great ideas, for 40 years. 40 years ago, people began thinking about gene therapy as a possibility. And now there's beginning to be some serious success, but in very small numbers and in very particular diseases. So there's still a long way to go to understand how genes make proteins, and proteins make healthy and unhealthy choices in the cell. We don't understand that process completely at all, or the gene therapy would be much more efficacious. Much less is understood about embryology, actually, and human embryology has been quite a puzzle. Many things that work really well in monkeys and mice really don't work at all in humans. Here, only 20% seemed even viable from the kind of test that they could do, which says nothing about the long-term outcomes, the children, the grandchildren, the next generation. So it's still very far from a safe, testable system, it seems to me. Okay. And I think we have to have a longer discussion about the ethical issues, about whether this is an appropriate use of technology, the best way to think about this disease.
All right, let's have that ethical discussion. I want to bring Josephine into it. If we could, as I understood from Brian there, you know, a little more research, a little more thinking, a little more discussion, perhaps get around some of the ethical issues. What is wrong with trying to find a potential cure to some of the kids out there? As Brian said, you go meet their parents, of people who are suffering from really horrible things. Is there not a case to be made looking back in history? I mean, every medical invention, or most medical inventions, has had some opposition, right? There are people who are against antibiotics and blood transfusion today, and eventually a lot of people have come around to saying that that's fine.
I think we have to distinguish very carefully between what is a cure for an existing disease in an existing person and what is planned here. So Brian's work in trying to cure genetic disease in children who've got these conditions, I'm all in favour of. There may be some aspects of it that need looking further into, but here we're talking about not curing the disease, but before birth, eliminating the carrier of the disease. So if you're using 2 embryos,
okay, but his point, if I understood it correctly, was and in the short term, let's prevent other cases occurring until we can find a solution for those who've already got these diseases.
Here's the thing. This technology is a very high-risk technology to solve the problem, not of the disease, but of how to prevent passing the disease on to the next generation. Because women are giving birth who have the disease, right? They're healthy enough to become adults, to get married, and to get pregnant and have a successful pregnancy. So the problem isn't their disease. The problem is that they don't want to have children that have a risk of a worsening condition, a worse version of the disease than they have. And there's very low-tech interventions there. You can adopt children.
All right, Laurie, hang on for a second.
Or you can, as we pointed out, you can use an egg of another woman and use that as a way to create an embryo without manipulating it to have your very own DNA.
Okay, Laurie, you're in the US right now. The FDA is looking at this question. Do you think it will approve clinical research?
It seems to me that the FDA is just the beginning of the kind of conversation that needs to happen. We have a President's Bioethics Advisory Committee. I would hope that they would take a look at this. Because of the long-term effects on subsequent generations, I would hope the Recombinant DNA Advisory Committee gets a chance to take a look at this. And I hope that the societies that do this work, the American Society for Reproductive Medicine and other societies that have said no cloning for reproductive purposes, would first have a chance to make the case that this is or isn't the reason to initiate cloning technologies. So there has to be a far wider debate than just the FDA, it would seem to me.
All right, well, it certainly seems like the FDA, I should point out, that's the US Food and Drug Administration. It does seem like it's taking its time looking carefully into this. But Brian, the question then is, even if the FDA doesn't approve this or slows down and really takes its time in looking into this, is that going to stop this research, or is it just going to happen somewhere else in some other country?
Well, I think that's also a concern. So, you know, one thing that the FDA and in the UK that the MHRA do is that they have very good regulation for clinical trials and very good governance and oversight of these procedures. So I think, you know, there is a risk that if we clamp down and say that none of this research can take place at all in either the UK, in Europe or in America, then it will go elsewhere. And will it be done ethically? Will it be done safely? I mean, clearly there are people who feel it shouldn't be done at all. But I would come back and say that, you know, in these mitochondrial disorders, they are maternally inherited. So that means that the mother always passes them on to her children. And in these cases, you know, the only other chance that they have of having a child who won't have the mitochondrial disease is adoption. Okay. So I think in this case, there should be a debate about this.
All right, we've certainly got a debate going here. Let's bring Josephine into this discussion. What do you make of that point? That if you try and shut it down, perhaps in the UK, the US, it's just going to happen somewhere else, isn't it?
Oh, well, I'm not very happy with the idea that the United Kingdom is a paragon of virtue in this instance. You know, we are the country that approved of the creation of animal-human hybrids, and we've done a lot of things that have got approval, but approval doesn't mean that they're acceptable or desirable. We have to underline the fact that this is not about curing disease, which has been pointed out, and certainly none of these technologies will be available in developing countries and other countries where, you know, genetic diseases are very significant. We need to find global cures that
hang on, Josephine.
Acceptable.
The stuff which I read was saying that the technique actually doesn't require a lot of complicated technology. And if you've got a fertility clinic in almost any part of the world, they could probably replicate at least some part of this research that's being done, if not do the whole procedure.
Well, I think it's always easy to say that when you've done it, but when you do see the success rates, the low success rates of this particular study, which is very much at its beginning, but what we don't know yet is what happens in future generations. I do know that in America there's an organisation called the Council for Responsible Genetics, a very secular organisation. They are very much opposed to this, because what we have to really underline is that you are changing the building blocks of human life. You are creating a new kind of human being, and that human being will have to carry that burden on into subsequent generations as well. I might be sounding alarming, but that is the biological reality. Yeah.
In 10 seconds, Brian.
Can we put this in perspective, though? So I mean, essentially, the vast majority of the genetic information is contained within the nucleus, 20,000 genes, whereas the mitochondrial DNA only contains genes for 13 proteins, 37 genes in all. So, in fact, you're not really massively changing the genetic makeup. They're not going to be a different person as a result of this. They may have changes in functionality.
All right, I'm afraid we are really out of time on this one. We're going to have to end it there. My apologies. There's plenty more to discuss and argue about, no doubt, though. But let's thank our guests at this point. Josephine Quantaval from Core Ethics, Brian Bigger from the University of Manchester, and Laurie Zoloth from Northwestern University. And thank you very much for joining us on this edition of Inside Story. As always, if you want to send us your feedback, email us your thoughts. InsideStory@aljazeera.net is the address. Thanks for watching. It's goodbye for now.